Is this working? Okay. So it's much like last time, 1 minute, and I'll give you a 5-second warning, and then I'll have to move on. But good luck. I'm sure you've all got wonderful work to show.
It shows it here.
Okay, hold on. I'm sorry, you practice everything, you hope it's going to work out exactly like you practiced, and then it does not. And so... Okay. Okay.
No, not working because okay, it's possible. Yeah, I think it's
working. Okay, just a minute. The audiovisual folks are going to help us out here. Is there... Great. Okay. Well.
Start? Okay. I'm Alina. I chose to do a PhD that is 50-50, computational and wet lab, which no one has told me before. I will feel out of place in both biology-focused and computational conferences. Hopefully some of you will join my poster anyways. So I'm here at EMBL Barcelona, and we work with a brain complication of malaria infections. And in the lab of Maria Bernabeu, we have this blood-brain barrier model that I perfuse with immune cells. And I do single-cell RNA sequencing. And what I saw, for example, was that there was an increased proportion of immune cells in the sequencing data when they were activated and stimulated with the malaria parasite before, indicating an increased binding of these activated immune cells. And yeah, if you want to see what happens to the blood-brain barrier cells if these immune cells come, then yeah, come to my poster. I'm happy to talk about it.
Hello, I'm also Alina, the second one here, and I'm also about single-cell RNA sequencing, so we have a lot in common. If you do single-cell RNA sequencing, you may feel like Homer Simpson up there sometimes. There are just too many choices for single-cell annotation tools that you could use from. And that's why in my master's thesis, I'm developing a tool called SE-Baptism that can run multiple annotation tools at once. So you as the user can just give your data to my tool once. It can run for you multiple annotations without you having to go through the hassle of learning various tools. And it will return to you the results conveniently in a single-cell experiment object with many added call data. So for every tool, you will get another column of metadata. And we will also provide you with some nice visuals, some that are convenient for single-cell annotation and also visuals to compare different annotations to each other. And I would love to talk with you about it in more detail at my poster. And since we're also in the development phase, I'm also still open for input. And yeah, thank you.
Hello. My mom wanted to name me also Alina, but my dad decided for Anastasia. So hello, I'm Anastasia. And I will I will present a poster about flow cytometry data analysis. So, as you might know, that flow cytometry evolved in recent years and allows us to measure up to 50 parameters across millions of cells in hundreds of samples. And as a result, you will get rich data set with huge discovery potential. But on the other hand, this greatness creates a great challenge for the analysis of this data with a conventional tool called FlowGel, limiting this efficiency a lot. To overcome this problem, we reviewed and integrated several bioconductor packages into a single workflow covering the entire life cycle of flow cytometry data, which we are happy to present here today in our poster.
So, hi, I'm Axel. GSVA at its core is a transformation of a data matrix, the rows of a data matrix, from, for example, genes to gene sets, leaving the columns unchanged, so that afterwards you can analyze your data on the gene set level. The idea and the package come from microarrays, actually, but it's a little bit surprising to see that it's still a favorable bioconductor rank, and the numbers of citations and downloads are still going up nowadays, which is, of course, because people are using GSVA and applying it to their single cell and spatial data. Hence, there's an ongoing effort called GSVA 2.x, trying to adapt GSVA to these additional types of data, including the algorithms applied to the data, as well as adding support for the corresponding bioconductor container classes. So if you have questions, what we are doing, want to see what has been done, what is still coming, if you have requests, we're happy to see you at poster number 6.
Hi everyone, I am Charles, and as we may know, glioma is a highly aggressive brain tumor, and tertiary lymphoid structures occur in 15% of cases, and they are linked to improved patient outcomes. However, little is known about their structure and their role. So in our study, on our patient-derived samples, we combine spatial transcriptomics alongside single-cell RNA sequencing and paired with B and T cell repertoire sequencing. We then analyze this data with quire and single R packages to obtain refined cell annotations whose signatures we map back to the spatial context, identifying the tertiary lymphoid structures. So our cross-modal approach shows how the lymphoid cells localize within the glioma tissue and tertiary lymphoid structures, and hence we are uncovering more about their biology and their therapeutic potential. So I'll be happy to walk you down more details about the clonal analysis and more information at poster 7. Thank you.
So imagine you find yourself in a situation where you're trying to analyze some data and maybe you know more or less what you want to do, and that may be something very specific, but you don't know what is the best way to do this with bioconductor. Is there even an easy way to do it? Was this the best package? What is the best function? This is what the bioconductor how-to collection is aimed at addressing. So a how-to document shows how to solve a very specific problem using one or more bioconductor packages. So if you're interested in this initiative, want to see what we have done, maybe want to contribute in the future, come to poster 8. I'm the next one. And if you don't want to analyze data, but you want to submit a package instead, you can come to poster 9. So here we provide an overview of the bioconductor submission process and also a bunch of tips and tricks for writing a bioconductor package and making the submission process easier.
I only have one poster, sorry. So this is work that we've been doing at the Department of Rheumatology in Zurich. For 2 years, I tried to understand what the biologists want to do, and they tried to understand what I can do. And after 2 years, I think we found a way to talk to each other. And so this is the effort of this. So we are using Cell Profiler. It's a tool that can extract morphological features from images that you see in step 1. And so it creates like a feature matrix of different things you measure about single cells, like the diameter and other things. And the conventional tools that are out there are usually a little bit hard for biologists to use and to kind of to make sense of, because the single features themselves, they are hard to interpret. And they also correlate very highly among each other. And so we tried to make it so that we can somehow make everything go into a volcano plot that you see at the very end, so that it's something that everybody's familiar with and easy to interpret. And so the CellPaint R package is trying to do that. Thank you for your attention.
So hi everyone, I'm Dario. So have you ever wondered about when working with single cell RNA-seq, when you're working with single cell RNA-seq, how well the clustering of your data is good, how robust it is. So if you want an answer to this question, you can try to use Robin, our Robin package. It's on CRAN. And the second version of this package allows to answer this question very faster. And we publish the results in this paper, so you can just check on it, or you can come to visit to my poster so we can discuss about how to use it and with your data, maybe. Thanks.
Good afternoon, I'm Elena. Why is another benchmarking necessary to evaluate the batch effect correction method for single-cell RNA sequencing data? Well, the previous benchmarking have some limitations, such as some of them tend to find the best method for all possible scenarios, but it's not realistic, and the consistency of evaluation metrics. If you are interested about this topic, please come to my poster, number 12. Additionally, I present you my R package called BatchChef, which contain a common interface for single-cell batch correction method in R and Python languages. Thank you for your attention.
So, hello everyone. I'm Fiona and I'm from Albania. So in my country, let's give you a little short information. We have recently put as the first AI Prime Minister. So we came with the idea, if an AI minister can be AI, why cannot we can use AI to understand and to help us the role of the gut microbiome on obesity. So that's what exactly we did here. We applied several machine learning models such as random forest, LDNA, and a new approach called Predomics to microbiome data from healthy and obesity individuals. So as a machine learning, Predomics performed as the best with a result of accuracy of 0.89. So the key step was using sharp explainability, which led us to move beyond predictions to see why the model makes the decisions. So I'm sorry. So, just like our AI minister promises transparency in politics, explainable AI gives us transparency in prediction, helping us identifying the microbial players related with obesity risk. So, thank you, and if you have any question, please ask me on the poster moment. Thank you.
Hello everyone, I am Francesca, and my project is about measuring others' activity. Others are enzymes involved in RNA editing, and specifically they catalyze the conversions from adenosine to inosine. Our method is based on transcriptional signatures that we have first defined and then tested, and now we are working on an R package that allows the usage of our method to get activity scores for others from expression matrices of bulk, single cell, and spatial transcriptomics data.
Okay, hi everyone, I'm Georgina, and as you may know, single-cell ATAC and single-cell RNA-seq have enabled the identification of cell type-specific features. Yet, there has not been any tool that actually quantifies this. So, we have developed SPICY, an R package that quantifies tissue specificity from multi-omic data. Essentially, it uses differential analysis results derived from commonly wide-used tools in single-cell pipelines to compute cell type-specific metrics, one for expression and another one for chromatin accessibility. This, together with Shannon entropy metrics, quantifies overall tissue specificity. Additionally, SPICY can annotate regulatory regions to their target genes, either through proximity or co-accessibility networks, which will help to unravel or construct tissue-specific regulatory networks. So, if you want to spice up a bit more your analysis, you can come and check poster 18. Thank you.
Hi all, I'm Hiru, and this is for the 3 or so people in this hall who work with transcription factors. But with the advent of ChIP-seq in 2008, we are now able to profile these protein DNA bindings down to a single base pair resolution. However, we do need a lot of background and a lot of input cells, and so we have much newer methods now, cut and tag, TIP-seq, and so on. But the problem is that all of these new methods compare themselves to the ChIP-seq. And we need some robust benchmarking method here, right? At the moment, all the methods really consider ChIP-seq to be the gold reference standard data set. But I introduced MotifPeaker, which operates on a very simple principle, which is that we take the peaks from the experiments, and the good peaks will have the motif of interest, and the bad peaks won't. Now, it's of course way more complicated than this, and if you would like to learn more, please come and check out my poster, even if you don't work with transcription factors. Thank you.
Hi everyone, my name is Hugo, and today my poster is named Writing Custom Linters for Package Developers and Their Users. But there is an alternative secret title that is Come Chat With Me About Static Analysis, because it's something that I'm really excited about and I would like to get you excited about this. So maybe first, what is static analysis? It's a way to analyze the code of your package or your script without running it. And it allows you to detect errors potentially or patterns of code that are not correct. So in a way, it's like some kind of super spell checker for code where you will detect like typos or maybe syntax errors or this kind of thing. In R, we have 2 main tools for static analysis. We have the linter R package, and then we have the RCheck tool by Thomas Calibera if you use compiled C code in your R package. And so my poster is just a way to trigger conversation about this, and also to encourage you to reuse the infrastructure that we have developed in the linter R package to create custom checkers for your own needs. And so I would be really happy to chat with you about how static analysis can make your code and therefore your life better. Okay. So
Hello, everyone. I'm Jingjing from Weihai, and I'm glad to introduce Smart ID, our novel approach for identifying markers for real populations from single RNA sequencing data. As we all know, real populations are sometimes quite important, and they are naturally existing in some tissues. However, most current methods would fail dealing with this imbalanced data due to its small number from the whole populations. However, by using Smart ID with 4 main steps, you can easily identify accurate markers for those real populations of interest. The first step for the input is always you need to provide a cell label as well as expression matrix data, either pre-processed or non-pre-processed. And then the data will be well and properly transformed into, scaled and transformed into an important score. And after applying a Gaussian mixed model, the accurate markers will be automatically selected for the specific real populations of interest based on the score. If you want to know more details about Smart ID, please come and visit my poster number 24. Thank you.
Hello, everyone. I'm Laureano. During my thesis, our lab faces a recurrent problem that there was no standardized way to manage capture high-C data, and even less to integrate it with other omic datasets. This requires in-house scripts and custom scripts, which make reproducibility very difficult. This is why during COVID lockdown, since I have a lot of free time, I started developing an R package for mainly in-house use in the lab. But at the end of my thesis, I realized that it has a true potential to be more broadly used. This is how HiCapture was born. It's an R package that loads capture high-C from many data formats, allows the integration of other omic datasets, also the comparison between datasets, and export the data to be used in other pipelines or visualization tools. If you want to learn more about it, come visit poster 26. Thank you.
So it was a cold and rainy day. It was in December 2018 in Munich at EuroBioC in Munich 2018. Johannes, the artist Rainer, who we have already seen, Sebastian Gibb, he's not here today, couldn't join, and myself were struggling, not with the weather, but with the software architecture for mass spectrometry. We had tons of ideas. We knew how to fix these issues, but kind of packages of the day didn't allow us the flexibility to do these changes. And so we did something that every developer dreams to do, start from scratch and do it the right way. And that's how the R for Mass Spectrometry initiative was born. So you have heard from Philippine about metabonauts, Johannes told about Spectra and Spectrify, Guillaume had a poster about PS Match, but there are many, many other packages for mass spec-based proteomics and metabolomics. So if you want to hear about the software or the community, please come and say hi on my poster.
Hello, I'm Leopold. So in our poster, we discuss about something that is sometimes overlooked when we develop a package, which is package optimization. So here we present some strategy to optimize package, and we also present a tool, which is VRR, to benchmark the different version of a package to see how optimization impacts the performance. And we will also talk about a case study on the Q-feature package. And as a sneak peek, we can see that our optimization led to a reduction of 95% of the runtime on a typical workflow. So if you're interested, please come to poster number 28. Thank you.
Hello everyone, my name is Mulka Anarim Ftouhi. We are presenting EuroBioC application, which is a visual platform for analysis and exploration of omic data. As all of you know, these kind of experiment and analysis requires some code, some coding skills actually. So EuroBioC actually offers the users an interface without evolving a code. And that's it. So we will be demonstrating our application using an end-to-end analysis from an end-to-end analysis of single-cell RNA-seq analysis from alignment to visualization. Sorry. So please visit us at poster number 29, and special thanks to Jérôme and Loïc. Thank you.
Hi, I'm Stefania from the University of Padua. If you ever work with cancer data, sometimes you may be interested in compute cancer signatures on your own data. Maybe you find some interesting signatures from the public literature and say, I would like to compute that signature on my data. But sometimes they often lack a computational implementation, and they are struggling to adapt them to your data sometimes. So we decided to collect the signatures from public literature, implement them, and add them to our package. So you can use SigFinder to compute cancer signatures on your data, and there are many ways to visualize the results. Thank you. Okay, I also have another package. Sorry, you were ready to go. No. I also have another package, a completely different topic, mitology. When you want to analyze your transcriptomic data and say, okay, let's do like a broad pathway analysis, you have many databases that you can use for pathway analysis. Sometimes they are even too broad, and the results may be confusing and not specific, and you obtain a lot of different results. So we were interested in my laboratory to have a tool that could allow you to perform large-scale pathway analysis, but focus on mitochondria. We collect mitochondrial genes and use them to filter public databases like Reactome, Gene Ontology, and focus just on what are the pathways working inside the mitochondria, and collect them inside mitology, okay? That allows you to perform large-scale pathway analysis just on mitochondria. And also there are some visualization methods to analyze the results. Thank you. Go to see my poster. Now I finished.
Great. Let's give a hand to everyone once again. I also wish to apologize for everyone, and I had to cut short just a titch. So I'm going to show you the schedule, and there's workshops, and you can choose which one to go to. Let's see. So. Is this one? Okay. So choose which one you want to go to. Is someone else? Okay. Yeah, so orchestrating spatial transcriptomics analysis with bioconductor is here. And then the other ones are down in the other rooms as listed here. Right.